Welcome to the spring seminar this semester. As you see, this is put on by Biology Department, the Science Club, and we have a few Science Club members here today. So if you're interested in joining Science Club, please see Brittany here or come and join us at one of our meetings. This is also put on by the newly formed Temple College Biological Research Institute, and so you'll be hearing more about this over the next few months. So this is kind of its first launch. Today we have a great speaker for us who's going to talk about some molecular ecology projects that she's done with some undergraduates at Southwestern. Romi Burks is here. She's got a BS and a BA in biology English from Loyola University, PhD at Notre Dame, and she's got a postdoc from Ohio State. Hard to say. Let's see what else. She does she's been at Southwestern University down in Georgetown for about 15 years. She's currently serving as department chair, I believe. Teaches a variety of courses. She teaches the freshman level course. She teaches conservation ecology, invertebrate ecology, research methods. She teaches a capstone course for seniors, and then she teaches a course in chocolate. So speak with Romi after the seminar if you're interested in that. Romi will talk a little bit here for a while, and then if we have any questions, we'll save those questions for the end and address those at that time.
Can you hear me okay in the back? Any problems? Usually that's not a problem. So I titled this Having a Shell of a Time. And I never thought I would care about snail shells. Who grows up and says, oh, yep, that's going to be what I'm going to study for, months or years? No one really. And the reason that I end up really focused on snail shells is that I used to have the idea that you could tell things apart by what they looked like on the outside. And over the last few years, I've learned that that's not true. And so you really have to go to the inside. And so about 5-ish years ago, I went down what I would call a rabbit hole into the areas of molecular ecology. So I'm a very basic trained freshwater biologist. I like buckets. I have my ecology students for methods right now. They're looking at little aquarium snails and buckets in my lab. That's great. I missed that. Most of my research now involves little tubes of stuff, but that's where you get the answers to the questions. So I'm going to describe to you some projects that I do with my undergraduates, and there's lots of undergraduates involved. I currently have 8 in my lab right now. They actually made me count on my CV. We've had 39 students go through the lab in the last 15 years. So they're all integrated in here, and I have a full CV if you want to know who did what. Okay, so that didn't work. Let's try this way. Okay, one of the things that I think is really important when you work at an undergraduate institution is to think about who your collaborators are. So I have a couple of collaborators that are outside Southwestern, and they have helped me really change my research agenda and broaden the questions that I can look at. But I don't have another aquatic ecologist really down the hall for me, like you would have at a great big institution. I don't have my colleagues in biology are great, I love them, but we don't have so much in common in terms of our research interest, although that's changing a little bit. And so when I look at who my collaborators are, they're really my students. And so, yes, I'm their mentor, right? So we have a mentor-mentee relationship, but they're also my collaborators. I count on them to be responsible for different parts of the research. I count on their expertise. I can tell you I can run a PCR. I can do that. I will tell you my students can run it 10 times better than I can, right? I can troubleshoot better than they can, right? But as far as bench work, they will kick my butt any day. Right? And so what we have done is set up a sort of timeline for undergraduate research. And so there's some initial recruitment, and that matters in terms of what are their motivations for doing research. Do they have time in their schedule? Is it a good fit? Are they really interested in snails? It's not quite as sexy as cancer cells, right? A little harder to sell. Often a lot of those projects start in classes like I'm teaching now, which is our sophomore methods class. And so right now they're looking at aquarium snails that are legal. I'll talk about the ones that we study most of the time, which are not legal. And they're asking some very basic questions. And so those will be just experiments that happen. But one of the things we're going to do with those experiments at the end is that we're going to filter the water out of that species, which is special. It's different than the other things that I've studied. And then we'll be able to extract some DNA and have environmental DNA from an aquarium snail. Then I can use that in a bunch of other things. So why the research methods things are, they're little cups in a bucket. They're not going to go anywhere. That's okay. I'm going to get something out of that at the end that can be used in my research students, and maybe those students will say, hey, I'd really like to follow this through, on a snail. So a lot of that starts in sort of this involvement in lab projects, whether in a course or within my lab, and then they'll eventually come to looking at independent research and maybe talk about addressing some bigger questions. We have also tiered some of that into paired projects, which I had not really done a lot of. On the cell and molecular side, it's almost essential. They don't have enough time to do all the cell and molecular things in a block. It's just, it helps to have more than one working on the same project. And so we've wrestled a little bit with, where's the intellectual independence there? But they all bring something different to the table. And then often I work with students after they graduate from Southwestern, because it's very challenging to do undergraduate research to the point that you can get it published while you're still an undergraduate student. And so a lot of my relationships extend past that. I just brought attention to, we just published this paper that was a session at the Ecological Society of America. It's written to other faculty members, but I also had my students read it, and they're like, oh yeah, it kind of sounds like you, right? Oh, you do some of these things. And so it was a good resource for them to sort of get the philosophy and the rationale for, yeah, you really have to have a contract, and you really need to send me Monday, Wednesday, Friday updates, because otherwise I don't know what you're doing, right? So there are lots of tidbits in there, and so I can give you the link if you're interested. Let's go this way. Okay, so I was trained in the lab of David Lodge. David Lodge is an aquatic ecologist that was at Notre Dame for a lot of years, and he's now at Cornell. And it's funny because I did my undergraduate on zebra mussels in Lake Michigan, and when I went to graduate school, that was the last thing I wanted to do, was to look at biological invasions. And so my PhD had nothing to do with biological invasions. However, it happened in a biological invasion lab. So I got the benefit of doing something very different, but it all has circled back. And it's funny because I turned down opportunities to work with snails when I was a graduate student, with crayfish. These are all things I do now all the time, and Dr. Lodge thinks it's funny. If you look at biological invasions, I've focused on invasive freshwater snails. There's a couple of reasons to do this. One is that they're rather understudied. So one strategy in undergraduate research is to find something that nobody does, or a few people do. Now, that can be good advice. It can also be bad advice, right? Because if no one's doing it, there's often a reason no one's doing it, because, oh, well, it's hard to do that, right? So you got to sort of balance that. I need to find my niche, or where are questions that I can ask. So I focused on invasive freshwater snails. Predominantly apple snails. These belong to the family Ampullaridae. They have both a lung and a gill. So they're interesting as an invasive species because they can stay out of the water for a fair amount of time. They're interested as an evolutionary link between land and water. So there's a vertebrate model for this, which is a lungfish. And so this is sort of the invertebrate model. And so they're not just an invasive species, they're an interesting group of species, right? And so it's really allowed my interest to go beyond biological invasions to things like species diversity, and what does it mean to be a species, and how do you tell them apart? So the other thing that has happened is that it has expanded into also having mystery snails. So mystery snails are within the family Viviparidae, and if you are viviparous, you lay live young. And so these are ones that lay live young. And so also freshwater snail, also some native in the US, but also non-native species. And these are not the same. Apple snails and mystery snails are not the same thing. However, if you go to PetSmart, you will see mystery snails commonly called, or well, you will see things commonly sold as mystery snails that are not viviparids. They're actually ampullarids. And so there's this huge opportunity, right, for public education in the aquarium trade as to, what are you selling? And this is also how these species get moved around. You buy it, you think it's going to stay small, it grows up very big, it eats things you didn't think it was capable of eating, but you feel bad for this snail. You don't want to kill it. So what do? You dump it in your pond. Bad idea, right? It floods, it goes, and it's out there. So there's a lot of room for what I would say, thinking about what it means to be a scientist and your responsibility in giving good information to publics and things like that. So we partner a lot with places in which we've seen invasions of snails to provide educational materials. Okay, so I'm going to talk about 4 projects. And so the first focus is going to be the most in-depth in terms of the molecular skills. It's about hybridization. And this has been the, I want to call it bane of my existence for the last 5 years. The paper is in review. It should go through. It's almost there. And I'm very excited about it, so I'll talk about that. And then I'm going to talk about, within one species, what is the within the species diversity and why that matters. And then we're going to switch. So these are all things about what a species is, how you define it, and how you describe it. The next set is going to be sort of about detecting invasive species. The new, it's not so new, reasonably new, technique called environmental DNA. And it's equivalent to crime scene shows. Anybody watch NCIS? If you've watched an NCIS episode, raise your hand. All right, you're all lying. There's like 10 of them. You can't, crime scene, you can't avoid watching one, right? But it's not, it's not quite that easy, right? But organisms shed environment or DNA material into the environment just like people. So you just have to know what you're looking for. So we'll talk about environmental DNA, and then we'll talk about coming up where understanding the distribution of these other mystery snails, these viviparids that are not apple snails. Okay. So in talking about a species, I have found this a good example, right? And so everybody knows an apple, right? So apple is a species. Malus domesticus is the original apple. It has been bred into a number of different varieties. If I didn't have the bottom picture up there and I asked you to name types of apples, right? We could go around the room, you could name a bunch of apples, right? So these in science are typically called subspecies or variety. It means that they can interbreed successfully, right? But they're all fundamentally the same species, okay? When you talk about different species, you're talking about apples and oranges, right? You can't crossbreed, as far as I know, an apple and an orange. Now, you can crossbreed some other weird fruits, right? It's all sorts of weird fruits, tangelos, right? But for thinking about the difference between subspecies and what we determine as species, the apple sort of works for me. And just like apples, both within the subspecies or variety show quite a bit of variation, so do snails. And so these are 2 species of snails, so Pomacia caniculata and Pomacia maculata. And so these are maculata. We'll talk about their name in just a second. Both of these species have been described as channeled apple snails. Channel means they have a groove in their shell, all right? Both of them are channeled apple snails. It's not that there is the channeled apple snail. There are lots of channeled apple snails. So it's a bad name, right? Because I don't know what that is. I can pick it up and go, yep, that's a channeled apple snail. But if I want to legislate against it, that's not going to work. I got to have a name, right? And so you look at and you can look at these shells, right? So these are all the same species, genetically. Right? And with Pomacia maculata, it used to be called Pomacia insularum. But a colleague of mine, Ken Hayes, went and collected a whole bunch of snails across Brazil. And when he did the genetics and did some statistics, right? And that's the line that you call it a species. Pamacia gigas, insularum, hostrum, amazonicus, and maculata are all the same things. They all look different from the person that picked them up and named them, but on the inside, they're all the same. So they have been synonymized. They all carry now one name, which should be Pamacia maculata, and the reason is it was named first. All right? So part of the science that I do is just to catch everybody up, right? It's to say, here's the modern understanding of what these species are. Now, there's a huge amount of variation in both of these species. They have big ranges, right? And so that also makes them a successful invasive species. So when we talk about apple snails, there is not one single species of apple snails. There are lots of apple snails. The main two that people read about or study are these two, Caniculata and Maculata. And that is where my original focus came. When a student at Southwestern brought me apple snails from Houston, my, third day on the job, right? I didn't know what they were. I had no idea. I'd never seen anything. And these are, they're baseball size, right? So 200 grams, right? Throw them. You can hurt somebody with these snails. Seriously. But I was like, what is that? Snails are supposed to be this size, right? Not this size. And at the time, Texas Parks and Wildlife was actually saying, oh, well, these are Pamacia caniculata. These are the Asian golden apple snail. And Asian golden apple snails are known to eat rice, so there was this big fervor over them. Turns out that they're not caniculata in Texas. They're all maculata, and they're not very diverse. But we didn't know that at the time. So in the 15 years that I've been doing this, total, from my first sort of introduction to apple snails till now, there's been a lot of change, right? And some of that is not yet trickled down into other communities. Oop. There we go. Okay, so I'm really just going to talk about 10 years, or 11, 2008. This is me in Uruguay. And so you're like, how did I get to Uruguay, right, with snails? Well, turns out in my PhD, I did some work in Denmark. And my mentor in Denmark was also mentoring a little few years down the road, an Uruguayan person that worked in shallow lakes, which is what I did before. And we just started talking. She's like, oh yeah, those are native in Uruguay. I was like, oh, well, that's cool. I should go there. And then, wrote some grants and got some money and went. And I was like, wow, these are amazing. And so what that did was to help build an international research program. And so research in Uruguay introduced me to what it takes to describe species, which is visits to museums, because you have to know what was there historically and what they called it, right? Are you finding the same thing or something different? Are you finding as many as they did or fewer? We also did quite a number of field collections. It's a little bit difficult to see in this slide. I'll show you some closer pictures, but all those pink things are eggs from Pamacia, from species in the genus. They lay their eggs above water. Remember I told you they have a lung. So the females crawl out and lay their eggs. That's important. Will be later on. We collected a bunch of snails. We worked in the museums and sort of created this program where I took students there for 4 years. And what that also did was to create a giant database for me, right? So I have enough samples and snails to keep working for quite a long time, right? So the last time that I was there collecting was 2015. So I'm still working on the material that I collected then. It takes a long time. Okay, so you might have some assumptions about snails, right? Anybody seen Turbo? Right, the little cartoon snail movie? No? It's really funny. Right? All right, snail goes to Indianapolis 500, right? Because he got struck by lightning and, he's powered, but he's slow, right? Snails are slow, right? But the research goes fast, and apple snails do get moved about very quickly, right? So in just a period of time, right, from early introductions in the sort of early 2000s into Florida and Texas, we now have apple snails, one species predominantly, right, throughout the Southeast, right? So they're in Texas, they're in Louisiana, Mississippi, Alabama, Georgia, Florida, South Carolina, North Carolina, although they've been maybe eradicated there, right? So they're not escargots, get it? Escargots. You guys are hard. I know it's in the afternoon, right? Right, but they're fast. They're fast. Okay, so there's some reasons to study apple snails. I've mentioned that they're a model of evolutionary history, right? So this water to land transition. They have a lot of interesting biological adaptations, including an operculum, which is the covering that the foot is attached to on the snail, and they can retract it, and they can hang out inside for quite a long time and under a various number of conditions. And they're particularly important in the US, apple snails overall, because they serve one species in particular, serves as a primary food source for an endangered bird of prey. So this is the Florida snail kite at the top. This is also a limpkin. There's also been a lot of work on crayfish as predators of apple snails. And I'm going to talk about there are both native and non-native apple snails in the US. And then we're kind of investigating a suite of a bunch of different predators that have been coming up. And what I've learned is that even when I say, oh, I think I know something about this, they surprise me, right? So I'm trying not to make assumptions anymore, right? That's been a big research lesson. So this is what they look like. So these are all Pomacia maculata, formerly Insularum. And so this is a female, this is a male. They're doing their thing, right? This is what happens when you take them out of the field and they stress out. They say, oh, this is my last reproductive opportunity. I'm going to lay all my eggs in this bucket. Right, so that's nice if you need eggs. You can see them on plants. This is at Armand Bayou. This is one of my favorite pictures I've taken. I watched the snail lay this egg clutch for about 45 minutes. Right, and so anyone want to guess how many eggs are in there? On average? 1 million. Nope, not that many. Anybody guess? No guessers?
50,000.
Oh, no, way high. 2,000. 2,000 on average. Looks like more, right? 2,000 eggs on average. They can lay one clutch, right? Basically a week, 10 days. Sort of depends on conditions, right? Summer, so they start laying in Texas in late April, early May, and they continue to laying through at least sort of mid-October. So you take that number of weeks, right, times an individual snail, times the number of clutches, times 2,000 eggs, times 70% of those hatch, and you get a whole lot. Now you're in the millions, right? So you get all of these introductions of little itty-bitty hatchlings into the system. Now, they don't all survive. Of course they don't. But it only takes, one or two, and you get more. So detection and preventing them from spreading is really, really key. Because once they're there, it's tough. It's tough. Okay. All right, so I mentioned native versus non-native apple snails. I just wanted to show them to you. Oh, I have a pointer. All right, so this is Pomacia pallidosa. This is the native Florida apple snail. They tend to be about golf ball size, right, compared to baseball sized, right? These lay about 20 to 40 eggs. Upper end kind of 80, all right, average of 2,000, upper end 5,217. The hatchlings are about 1 millimeter. These are about 3. And so they can compete sort of at the size range, but not at the numbers game, right? So they lose out in the numbers. And if you look at overall, Ampullaridae, they lay eggs. They have this round shell, and they're vascular consumers of plants. These guys will also eat a lot of algae. You can see it on the shell. These guys like real plant material, right? And so that's them eating lettuce in my lab. And then overall, if you look at non-native apple snails or apple snails, they can have large native distributions. So Pomacia maculata is native from about the Amazon down to Argentina. That's a lot. That's a big area. Right? They get large sizes, as I mentioned. They can hold a number of parasites, and so they have public health implications, and they're involved in the aquarium trade. All right, so the thing about molecular work is it basically all is the same techniques, right? So we basically do the same thing, more or less, for most of the questions that we answer. So we have to get the snails and dissect them. And so we get them either live, we collect them, sometimes they come in the mail, sometimes we get them from museums, and we dissect them and take a little piece of the foot And so that's the muscle. And we try to do that because it doesn't have as much glands and other things that can interfere with DNA extraction. And so then we'll extract them. We've done this in a couple of ways. One, we've used a kit, right? So everything's in the box and you follow the directions. And then lately we've been using, so this is supposed to give you really nice sort of clean DNA. Lately we've been using chloroform. And chloroform extractions give you bulk DNA. They give you a lot more DNA, but it's not so clean, right? And so there's some sort of balance between these two things, and it depends on what you're doing with it. And so we spend a lot of time troubleshooting in the lab. Then we go to PCR, so polymerase chain reaction, where we have particular targets. Our targets have focused on really three that I'll describe, but one of them is CO1. That is the cytochrome oxidase C gene, and it's often used for barcoding, like when you go to the supermarket and it says, okay, I'm buying, waffle mix, right? And so a barcode, like a species, you could scan it and say, this is what this is. And then 16S is another ribosomal barcode marker, and we've had to, CO1 works pretty well for apple snails. It doesn't work for mystery snails. Don't ask me why, I don't know. But we've had to change a lot of procedures to accommodate that. And then we go and we take its sequence and do the analysis using a bunch of software. So another piece of advice for students is take computer science, right? Take computer science because a lot of the data analysis that you're going to do involves computer programs. Okay, so we do this, we edit, and then sometimes we get good sequence. Good sequence is easily called. Bad sequence, not so much, right? And then we take that and we do this series of additional analysis that will tell you what species it is, and I'll explain that when I see it on the slides. Okay, so this is where my collaborators come in, and so Ken Hayes his dissertation was to describe the diversity, and you can see that there are some Pumacea that are not yet named. Right, so they're genetically distinct from these other ones. So we focus on maculata and caniculata. I'm going to tell you some things about Pumacea species as well. And there's also other genera. And so Marissa is in Texas. That's a non-native species as well. Philippinea and Acyllenia are both in South America. And so we've used this to ask questions of cryptic species and hybridization. Okay, so now I'm going to show you data about hybridization. Okay, so let me introduce 4 species in South America. The first is maculata, that's the one we also have here. The other is caniculata, that's the famous one that most people study. This is Pumacea species, hasn't been named. And this is Pumacea megastoma, means big mouth. So if maculata are baseballs, megastoma are softballs. Right? And so we've looked at these different questions, and we were particularly driven by publication of this paper that suggested that there were hybrids of maculata and caniculata in their non-native range of Japan. Now, they were able to investigate using molecular tools and suggest hybrids, but the difficulty with hybridization is that you can get mixed alleles in different ways. And so, And so you can have a scenario where they just persist in one population, right? That's the orange line. So you have a speciation event and you have the alleles that are sent that direction. You can have them persist in both lineages, and this is what they saw in the Japan population. So it's incomplete lineage sorting. It's not all moving into one species, but it's shared. Or you could have the orange, where you had it in one species, but then it comes back into the other through hybridization. To detect this, you have to have both mitochondrial DNA as well as nuclear DNA, right? And you have to have instances where you have overlapping populations and non-overlapping populations. And this is something they didn't have in Japan. So they weren't able to resolve this question. And so we have, which is very exciting. So this is a very busy graph, but I'm going to show you what it is. So this is a map of South America. This project was primarily centered in Uruguay and then with collaborators in Brazil. And so these are haplotypes. These are the percent of identities between maculata and caniculata. So this is mitochondrial. This is what we would call them. This would be their species name. So you can see that maculata, right, in the hatched is all the way through Brazil. They're all mitochondrial. They're 100% maculata, right? All those hatched orange ones. Here we have a little bit of caniculata at this site. And we have basically a mix right here. And then we have some sites where we have only caniculata, the blue hatched. So those are identities. Then we go back and we look at nuclear DNA at those sites. And so one is, okay, we have both of these species, but look, we have caniculata DNA in the nuclear, and we also have mixed. That means they have one allele of both. One parent was one parent was the other, right? And you see mixed here, right? They're not solid like they all are right here, no hybridization. And then there's this really annoying dot up here, right? There should be no hybridization up here. This is weird. These are all maculata. They were thought to be maculata, but hey, they have nuclear alleles of caniculata and mixed ones. What happened there? All right, there's a fishing village resort right there, and people from Argentina vacation there, and they take snails as bait. That's what we think. Okay, so this is the phylogeny that sorts that out. So the tree that you see is mitochondrial, so all of the orange are maculata. If there was no hybridization, the nuclear would match 100%. All of this would be orange. Right? But there's some blue there. So we have caniculata males mating with maculata females. Here's the blue. These are all caniculata by mitochondria. If there was no hybridization, this would all be blue. You see some orange. That means we have maculata males mating with caniculata females. So it's bidirectional hybridization, and then we can quantify how much of that happens. This was an extraordinarily large amount of work. Done primarily by one student, 2 students, right? Super excited, and yeah, I'm having a party when this paper is published. Okay, so the other student that helped on this project was not necessarily interested in hybridization, but interested in overlap between species. And so what she did was to identify species and then take all of those that identified as caniculata and look at the variation in the gene within all those caniculatas, okay? And so this happens with a haplotype map. And so here's how you read one. One step, here to here is one base pair change, right? So it's a C in one and it's a G in the other, okay? So between here and here, 1, 2, 3, 3 steps, 3 changes. Okay, the different colors are the different sites where we collected them. The number of circles are the number of haplotypes, and a haplotype is a unique genetic sequence within a species, right? You may have several snails that have that haplotype, but it's unique. There's 1 or 2 differences. A black dot is we haven't found that specific haplotype, and the size of the circle is how many samples are there, okay? So it's a little hard to understand. I'm going to show you a very simple one. Here's a very simple one. This is Pamacia maculata across the southeast. This is in the US. So how many haplotypes are there? 1, right? So number of haplotypes. 1 big circle, right? 1 little circle. There's 3, there's 4, there's 5. There's 5 haplotypes. How many individuals are in number A? Well, that circle is 10, and so I can extrapolate that's probably about 50 snails. Okay? Where do you find haplotype A? Well, you find it in South Carolina and Georgia and Louisiana and Alabama and almost everywhere. Where do you find this one? Only in Texas and Louisiana. Right? Where are they closely related to? Ones that are from Argentina, right, versus ones that are related to Brazil. So we have 2, and we get these from GenBake from other sequences. So we have 2 introductions here. We have some snails coming from Argentina, and we have some snails coming from Brazil. All right? And so so not a lot of diversity within the invasive population of Pamacia caniculata. Okay, now don't blink. Here we go. Okay, this is caniculata in its native range. So different species, but you can see that in this case we have 27 different haplotypes instead of 5, right? And they're clustered in sort of a regional sort of background. So this is Argentina. Over here, lots of diversity at the different sites. All of these are missing because there's a big river in the middle of that we can't sample, right? And then we get to sort of the Uruguay populations that are different and more clustered, okay? So all of these are Pamacia caniculata in terms of their identity, but they show some genetic population structure. And that can show in landscapes and how they get moved around. All right, now, this is the really exciting result of this work, this is Pamacia species. This species is a cryptic species to Pamacia caniculata. And so before this work, everybody called this caniculata. All the museum samples call this caniculata. That's what it was called. But it is genetically different, and it has a huge amount of genetic variation within this species. So this has 92 different haplotypes, right? So again, 1 base pair different, 2 base pair differences, sometimes up to like, I don't know how many is between that, 30, right? Big differences, although they all clustered together. And so having this wide range of genetic variability in a marker that is highly conserved, right? CO1 you need for breathing. You don't have it, you die. It's highly conserved, right? Just says, hey, look at all that adaptability in there, right? And so now we're starting to see the Asian distributions come out and identify some of these nondescript species that they thought were caniculata or maculata. Okay, so just to recap that, hybridization does occur in the native range. Other species can be invasive, so that Pamacia species is invasive. It's spread everywhere, right? But in the native range, maybe also in other places. And then, it's very hard to tell some species apart from the outside. Got to look inside. And then we continually change what we call things and describe things, and you just want to keep up. Okay, so now I'm going to shift to detection work using environmental DNA. And so we have used apple snails as a model in which to investigate questions about environmental DNA because we've had a lot of genetic material for them. So this was possible because we had genetic reserves of extracted DNA. You have to start somewhere with environmental DNA. You have to know what you have to start. If you want to find something, you have to know what you have to start, right? And so environmental DNA is really critical for focusing on the spread process. So introduction and spread is where we are interested here. And my students, Chelsea and Lauren, they like Edna Mode, eDNA, get it? If you search eDNA on Google, you get Edna Mode, not environmental DNA, right? So it is the extracellular residual DNA shed from an organism in various abiotic environments. So it can be in soil, it can be in air, it can be in water. We work in water. And so you have, most of the work has been done on fish or frogs, and so you have all of this, right, fish business coming up. They're moving around, they're getting off scales, slime, right? It matters how long, it matters what temperature, right? All of these different conditions. Understanding how these work is called the ecology of eDNA, right? And so ecology is looking at how things change in a distribution, right, in an environment, and that's also what we're doing within environmental DNA. And so our primary research objective when we started was to say, all right, can we look and find Pamacia maculata using environmental DNA? And so we know they crawl out of the water, and we know they lay these pink egg clutches. So once they're there, they're pretty visible. I don't need to do all this molecular stuff to say, oh, there's apple snails there. I can see pink. However, if I look in the winter, no pink, right? If I look in the really hot summer, sometimes no pink because they're too hot, right? They stay in the bottom, right? And so what if I didn't actually see the pink? What if there are some snails there? How many snails need to be there for me to see pink? I don't know the answer to that question, right? So environmental DNA is a way to both confirm field sightings and also investigate places that you might think have apple snails. So if you recall, Houston was underwater for quite a long time, and that moved around a bunch of snails, right? And so we might not see them, but we could test the water. So the first thing that you need for eDNA is you need primers. You need little pieces of DNA that attach to the DNA that you want. In contrast to when you look at species, CO1 is 658 base pairs long. You really need all 658 if you want to do a good job. eDNA is in bits and pieces. It's like you chopped it up with a knife. And so our base pairs in this case are 80, 100, 123, 150. These are our regions that we're targeting. They're not big. That creates some problems we'll talk about later. But what we did first was to create some primers to see if these will work. And so we wanted to say, does detection vary among the primers? And what primer can we use in field sampling? And so here's our invasive crime scene, Cullinan Park, which is in Sugar Land. There's known species right here in White Lake. That's a picture there. But there's no apple snails in this part of Oyster Creek. And so we sent students out to sample and to see if we could use our primers to say, yes, apple snails are there or not. Very simple. And so we collect the water samples. We can confirm snail presence, but we really just need water. And then we filter the DNA, right, onto a special filter. And it goes in a little tube and gets extracted. And then we do quantitative PCR. And so what this does is it tells you where along in the cycle of PCR you detect that molecule. The earlier that you detect it, the more there is. You only need one molecule for DNA to work, or for PCR to work, right? So if you have more, it works faster. Or not faster, earlier, right? And we do this on a qPCR machine. Okay, so here are the results of that study. And just to, these are the three sites. These two sites are the lake. This is where we didn't think there were any snails. And so the different colors are the different primers. You can see that we had sort of widespread detection here. Some primers were more consistent than others, right? But we did get some hits here. Now, detecting eDNA, you should know coming in, is kind of a crapshoot. You have to replicate it a lot, right? Because you're picking up 250 milliliters of water out of this giant thing, right? And you're saying, oh, I'm going to get that, right? So we were really excited that we even got anything, and particularly that we got snails where we didn't see them. Okay, and this is you can see sort of the variation in the replicates. And so again, this is a log scale. So some of them are more consistent, some of them vary. If you're going to work with eDNA, you have to come to terms with it being variable. It is the worst data I've ever seen in my life. You know, worst in a qualifier, right? Because it's just variable, right? And so if you really want to find things, you really have to replicate and think creatively. Okay, so the other thing that we're doing with this is to say, okay, well, you know, we optimized what we thought was Pomacia maculata, but it turns out that it wasn't quite enough, that running it at 60 degrees in an annealing temperature actually amplified some other things. Some other Pomacia, okay, because again, we have this DNA that we can use, and this was all tissue derived, but we want to make sure that we're not, you know, amplifying Mysterysnails or Campyloma, just a different viviparid. We want to really target, you know, this maculata. And so we're doing a lot of optimizing of both tissue and eDNA as we have it across a temperature gradient. And so that's another one of these eDNA projects. And it all really comes back to looking at the ecology of eDNA, because it's a great tool and it's being widely used without us knowing a lot about it, right? Phylogenetics and systematics and how to identify species has been around for 400 years. This stuff's been around for 10, right? And so we don't know what happens with the origin of the material. Is snail DNA different than fish DNA? I don't know. Snails have a lot more slime, right? What about the state? What kind of filters do we need to detect different materials in different environments? Where is it going? How fast is it decaying? What if you put antimicrobials? What if you put antibiotics, right, in the water that are coming through runoff? What's happening to the eDNA? It's being degraded by bacteria, right? What if you put in something that affects the bacteria to degrade the DNA? We don't know what that does. We have no idea, right? So this whole field, and this is why I talked about getting a niche, is that my students can do anything they want, as long as it has an apple snail or snail that we have DNA for, right? And fall somewhere in this world. Because what they're going to do is new, and we don't know. And we have a model that we use to do that. And so the last project that I just want to speak on is where we're going to take some future work that I've learned from all of this and are now sort of channeling into these mystery snails, which, you know, have a horrible name, mystery snail. It's a mystery as to what you get. And so there are at least 2 species that are in the US, Cypangopaludina chinensis, guess what, it comes from China, and Japonica, that one comes from Japan. And so very similar to maculata and caniculata within Pomacia, they look very similar. And they're also not well studied. Again, freshwater snails. And so we got into this game by chance. So this is my student Avery, and she did a lot of behavioral work with Pomacia maculata, and I sent her out with Bianca to collect some snails in Houston, and lo and behold, they found Japanese mystery snails there. Didn't know what they were, no idea. Had another collaborator, Russ Minton, that said, oh yeah, those are Japanese mystery snails. And you know what, they've never been found in Texas before. And so there's a publication. That was awesome. And that was literally running around and finding 2 snails. Okay? Now, if I knew that they didn't, that the genetics was not as easy as this paper, maybe I wouldn't be doing more, but it's okay. And so we were interested in, like, okay, how much of these are being confused, right? What is our understanding of what a species is for these? So we've looked at the distribution of Chinese mystery snails and Japanese mystery snails, and you can see there's our dot. So they're not anywhere close, right? And so if you look at Japanese mystery snails, we do the same thing to get this DNA to do that. Okay, that doesn't matter. All right, so we have sequences of 48 snails. They come together to create 48 unique sequences that we can then put in a phylogeny model, and this is what we get. So this is a phylogeny of mystery snails, and so we use an outgroup. So we have sequences from caniculata. They're not mystery snails, so that's a nice base. And then we can say, okay, well, the viviparids we get from GenBank, they fit really nicely there. And then our Japanese mystery snails are up there by themselves. And wow, we got a couple unknowns. That's always fun. And then look at Chinese mystery snails. Huge amount of variation. Very similar to what we would see in Pomacia species. So these freshwater snails that are invasive, that have wide within species variation. And so we're interested in what that means, right? And you can take something like mystery snails, and we have a very limited sample size here, but that dot right there, so haplotypes here, right, are 7, 1, 2, 3, 4, 5, 6, oh, I guess that's 7. 1, 2, 3, 4, 5, 6, 7. Yes, 7 haplotypes, bunch of different sites. Those are the colors, right? So this haplotype is found in 3 sites. And so Texas is the red one, right? It's also found in Pennsylvania and it's also found in South Carolina. These places are not close to each other, right? So where are those coming from? They're coming from the aquarium trade, right? And so we can say, if we say, hey, you should really not be selling these things, right? And eventually, maybe you could track the source. That's ambitious. Okay, so I am trained as a very basic researcher. I like basic questions of how science works, what things do, and that interests me, right? Now, I've really nested all of my work within these very applied research connections. So whether they're on invasive species, whether they're on public health, because freshwater snails carry a number of parasites. This one is rat lungworm, really terrible, goes to your lungs, then nests in your brain, causes encephalomyelitis, swelling of your meninges, nasty, right? Not good. And these are increasing. So you've seen things about, in the news, measles, right? Measles is increasing. So are zoonotic diseases, right? And so there's a public health concern, and then there's a education concern, right? That people don't know enough about these species and what to do with them, and so we want to make sure that they get reported. And so if you ever see snails that you don't know if they belong, right? Contact some professionals. Type in, snail ecologist Texas. Hopefully you get me. Maybe you get somebody else. It's okay. Ask them. All right, report to the USGS. All right, and you put them in a jar of alcohol. It can be isopropyl alcohol, right? You're not going to kill, right, the whole population. Not likely, right? And then you get it correctly identified. And so anytime anybody finds a snail, and I get some random snails in the mail sometimes, what is this? I'm like, okay, I'll figure that out. And so highlights of the research program is that we've really focused largely on a family of Ampullaridae. This is branched out into viviparids. That understanding biological invasions is challenging and has several steps along the way. We focus both on detection and spread. Common names are terrible, right? They're easy for people to confuse. They're impossible to legislate. People just need to name things what they are. And diversity makes for a possible good source of invaders. So we've seen this with both Cypangeopalidina chinensis, the Chinese mysterysnail, and also within the cryptic species and maculata. Environmental DNA, they like to call themselves molecular detectives, right? The eDNA mode. And there's lots of interest in the ecology of eDNA, or eDNA as a survey tool. And there's lots of applications and jobs for people that can understand, right, what a detection really means, right? Does it mean that it's there? Was it there? Was it there 2 weeks ago? Was it there yesterday, right? Are you going to find it again tomorrow? Who knows, right? And so I've sort of built this model again on apple snails, and a lot of it I'm extending to mystery snails. And one thing that I will tell you is that research leads you in many directions. And I've been fortunate to be able to go down many different roads, and that's fun. So I'm happy to take any questions. That's it.
Any questions?
How much did your qPCR machine cost?
The qPCR machine, I think, was like $35,000. Yeah, it's a pricey little piece of equipment. The plate to calibrate it is $700. You can only use it 2 times. And it's very funny. You have to spin it, and we don't have a plate. We don't have a centrifuge that spins plates at Southwestern. So I had to take the little plate to clean the PCR machine to UT to spin it to pain in the butt.
So is it imperative to do the qPCR in your early stages of doing eDNA research, or can you?
eDNA is only done through qPCR. In a regular PCR, you're never going to get enough material to detect it. You need to use a quantitative, polymerase. There we go, like Cyber. So TAC won't do it. It's too small. Super small. Yes?
You You said that in North Carolina that those apple snails may have been eradicated, that invasive species. How do they go about doing that?
So in the case of North Carolina, they copper sulfated the ponds.
Oh, okay. All right.
So they were on utility property, so they were owned, again, this is my understanding and I haven't checked in like the last few months. They were detected on a utility company property in these sort of reservoir ponds, and the not the city but the county owned them, and so they had the realm to copper sulfate them,
Okay.
and so that will kill the snails and everything else.
Everything. Okay.
Yes. Now, if they didn't get them all, or they were flooded in from somewhere close, they'll just repopulate.
So the eDNA is incorporated into bacteria, or just bacteria just digest?
No, they just degrade it like anything else. So it's not incorporated into, it's just broken up into many more parts. So think about, a 10-piece puzzle, right? You can do that one really quickly versus a 1,000-piece puzzle. And eDNA is the 1,000-piece puzzle. Yeah.
From the time that you run your sample through the filter
Mm-hmm.
to the time that qPCR is done, how long does that take?
That's a good question. So run it through the filter, and then you have to extract it. Extracted comes in 2 parts. And so you need to extract it, needs to incubate. So it's a 2-part, it's a 2-day thing. The first part takes my students for our routine lot is about 12 samples at a time. I don't like them doing really any more than that. And that's about 2 hours of their time on the first day. It's about 3.5 on the second day. So that will get you total genomic DNA. So it's all DNA that was in that sample, not just apple snail, right? And then qPCR is pretty quick. So all you have to do is plate it. And so you put it on a plate, and that takes, it takes about an hour for them to plate. It takes the machine about 2 hours to run, and you have data. It's magical.
Is that the 2 and 3 hours between the samples, is that active time? Or is that you're incubating and it's not hands-on time?
The hands-on time is at extraction, and then you plate it. That takes about an hour. Then you put it in the machine, and you leave it, and you can go do whatever you want. And you can come back, and the data's there in 2.5 hours.
So what kind of plate is it? Is it a microtiger plate or is it?
It's a 96 well plate with conical tubes.
Similar to a microtiger, but different. And it's
Yes, can't be flat because, and each plate is specific to each machine. So you have to get the plates that the machine can pick up and read. You can also do it with little tubes. So, but most of the time, it's easiest to do it on a plate because there's 2 things you can do with eDNA. One is you can use it simply for detection. So you could say, I have these control samples, I know what they are, and I have an unknown sample, right? And I want to see what that is. And then I leave some non-template controls so that I don't get any contamination, right? So I have basically 3 types of samples, and all of those have to be replicated. And usually they're replicated both at the filter stage, so say 3 samples of site A, and then they're in triplicates on the plate, right? So 3 times 3 is 9 times however many sites times the controls times whatever. It doesn't take so long to fill up a 96 well plate.
right.
And you want some space in between there because unlike regular PCR where you may see the contamination if you drag your pipette across the tubes, you will definitely see eDNA. So you have to design a plate so that you can pipette around. You cannot take your pipette over ever an eDNA plate. So there's some puzzle, kind of like Tetris, right, in designing the plate as to where your samples can be. So I didn't add plate design, that's another hour. Oh, so then the other way that you do it is you actually have a standard curve. And the standard curve would tell you how much eDNA you have in there. And so that standard curve takes up 15 wells. So 5 points, multiple dilutions.
So the point of quantifying eDNA quantity, is that to predict or estimate abundance?
Eventually, yes. So there are a few people that have some reasonably good data on the relationship between eDNA copy and field abundance. Most of that work's being done out of Japan with fish. It's really hard to get that equivalent. The reason that you would want to quantify the amount is if you're looking at what external factor is influencing that, right? Because just a hit is going to happen anytime it hits a molecule, right? So is it just a random molecule that's out there floating around that happens to hit, right? Or do you have lots of molecules there that indicate that indeed your organism was there? So it's a probability game. Yes?
So the cytochrome oxidase gene is in the mitochondria, right?
Yes.
So if you inject, like, let's say, antibiotics, like you were saying, to the snail, can you create new varieties of snails?
No, because you wouldn't affect the whole genome. No.
They probably die or something.
Well, anything, so, it's redundant. The CO1 gene does have variability in the toggle position, in that third position, but anything that you influenced prior to that would change a protein code would not work. Yeah, it would die.
So if you don't have a qPCR machine and you collect eDNA and get it analyzed, are there companies that you can pay to do it and send your data to you?
That's a good question, and I have not had to do that, so I'll be honest, I haven't looked into it. Okay. I will offer we have a qPCR machine 45 minutes down the road.
Okay. Yeah, I don't know what your load is and training and...
I mean, my undergraduates train my other undergraduates to do it. So it's regular PCR. So I do regular PCR without any pre-made master mixes. So regular PCR for me is my students adding buffer, polymerase, primers, what else is in there? Salt, other stuff, right? So it's a lot of pipetting. So they're, likelihood of error is great, right? With qPCR, it's not making the reactions. The reaction, you make a big master mix. It's the consistency of pipetting 20 microliters into your well. And so it takes practice, right? And you will get...
So you started practicing up here just in a micro titer plate of pipetting 20 microliters and checking the accuracy of their pipetting.
That's a good start. So you want your triplicates to be very consistent, right? The problem I have found with students is not triplicate consistency. I mean, they can actually get quite good at that. It's awareness of what they're doing so that they're not crossing the plate. So contamination is... It's not, so Matt Barnes is my collaborator on this project at Texas Tech. His phrase is, it's not a matter of if you get contamination, it's a matter of when you get contamination. Like, it will happen. Yeah. And so what are you going to do when that happens? So eDNA needs to be in its own place. It needs to be not extracted. So we've had to move all of our extractions. They used to be done in my lab downstairs, but there's too much free-floating snail DNA in there. Right, so that it gets contaminated. We actually just bought a solo chamber, put it on a little cart, wheel it around PCR thing. It's like $4,000 just for eDNA, so that it's not in any hood. I mean, you sneeze on it and you get contamination. Now, having said that, you can get past it. I don't think I realized it when I don't know. So ignorance is bliss until you're like, oh yeah, well, if I would have known that, would I have ever gone down this pathway? I don't know. Good question. But now I'm already down the rabbit hole, so what do? I just learn to deal with it.
Right. Okay, all good things to know.
Yeah. But the students are, they can be really good. And they can learn it. So there's some naturals, right? They have, good hands. But they can also learn. I've seen both.
Absolutely, yeah, I have to.
I've got a question. It's not about eDNA. So it's about, you mentioned, it's always been curious to me about trying to figure out what is and what is not a species. You talked about maculata, and there were formerly, 5 different species they thought they were?
Yes.
You guys looked at them genetically.
My collaborator Ken Hayes did that as his dissertation.
Okay. That's okay. And so, so you weren't involved with that research?
No, I was not directly involved in the rename. I supplied samples for him, but no,
Okay.
that is his area in dissertation work.
Oh, okay. I see. Okay. Well, my question was kind of about that,
But I'm likely to be able to answer it.
Okay. Well, so what? It looked like you showed a picture of the snails, and there were a lot of different colors of the shells and sizes and things like that. And so, how much of that is just phenotypic plasticity within a population, or how much of it is like based on geography, like different?
Both and both.
Okay, both.
So within, if you look at intraspecific variation in maculata and caniculata versus interspecific variation in the genetic markers that Ken has looked at, and he has not only done CO1, he has done 16S, H3, N5, I don't know what the N one is, 6 or 7 different markers.
Okay.
Within variation within the species is around 2 to 3%, which is a lot.
Okay.
But between variation is between 8 and 10%, or sorry, between species is about 8 to 10%.
Okay.
So when you draw the lines phylogenetically
Yeah.
through lots of bootstrapping and multiple phylogenetic analysis using both Bayesian and traditional maximum likelihood approaches, you see this gap between intraspecific variation and interspecific variation.
Okay. Is there still some degree of like arbitrary cutoff? I mean, I know because we're defining the boundaries. I mean, could it be that some other people would say that some of those populations could be their own species because they're on their own independent evolutionary trajectories? Or?
Absolutely.
Okay.
Yes, it depends on who makes the. So there's lumpers and splitters, right?
Okay. Yeah. Okay.
Yep.
That's really what my question was all about.
Yes. So Ken is a lumper,
Okay.
right? And so these are all genetically the same at his analysis of this, and therefore they are all the same thing. It doesn't mean that they're 100% identical in all populations, and certainly there's a huge amount of variation. But I would say generally he's a lumper. So as an example of that, so I gave you there's 3 other genera within Ampullaridae, within New World Ampullaridae, and those are Marissa, Asolini, and Philippinea. And I expect in the next 3 years they'll all be Marissa. Right, and then Asolini and Philippinea will become a subspecies.
Oh, okay. Thanks.
Based on that work. They all look the same on the inside.
What's Marissa? What's the common name of Marissa?
A ram's horn snail.
Yeah.
Bad name, giant ram's horn. There's lots of ram's horn snails. It's in the San Marcos River. So, that's what I did.
All right. Well, if you have any questions, come on up. We can continue talking up here, but thanks for coming.
My pleasure.